That's right, I have to do it from here. All right, bad joke on the first slide. I'll let you look at it and then won't say a word of it. Let's start with, I can't remember what this is. Oh, yes I do. Yes, this is actually a beautiful picture. I'll put this on at the end of the lecture. It's not totally relevant. Okay. Without using your clickers, because I still don't know how that works, tell me approximately half of you should have had a fungal infection, be it candidiasis or athlete's foot or jock itch or something like that. We don't have to volunteer now. It's against HIPAA violations. So you don't have to answer this. But the bottom line is fungal infections are more common than you want them to be. Okay, and today we're going to talk about morphology. We're going to teach you that boring classification. We're going to teach some cell biology. We're going to tell you about how fungi come in different, the same fungus comes in different flavors, dimorphism or polymorphism, pleomorphism. Tell you a little bit about diagnostic techniques. I think what I'm going to do is go through a little bit of the mechanism of antifungal drugs because Dr. Howland may not be able to give his lecture today. So I'll just run through a very, very cursory stuff on that, but you should defer to Dr. Howland's lecture even online, and Dr. Garrett will tell you about that later. Okay, this slide I'm not going to read to you, but the bottom line, nor am I going to read the content objectives to you. They are for you to read. Okay, here we are, fungi. What are we talking about, fungi? And this is coming out in the projection, right? When I point to it, we're talking about yeast. These are single cells that grow via budding. We're also talking about molds, which have hyphae and grow into these mycelium shown down here, and you've probably all seen them growing on your food. And we're also talking about mushrooms. And mushrooms are simply a organized form of molds. If you look in the microscope, you'll see lots of thin tubes making up these beautiful mushrooms, which are, of course, delicious, but also highly poisonous, and you'll see in some cases pathogenic. Now, my favorite mushroom in all lifetime is a puffball. Okay, now I once calculated that a puffball, and this is the daddy of all puffballs, if every one of the 10 to the 14 spores that are in this actually germinated, it would fill the Mediterranean Sea. And I tell you this because fungi have a propensity to grow. And that propensity to grow can make them highly pathogenic depending on the environment of the body and what the body's defenses are against that. Now, in fact, this used to be the daddy of all pea balls. It then transitioned to the ditty of all pea balls. Now it's simply the ditty of all balls. Anyway, as doctors, you're going to have an important job getting rid of the astonishing yeast menace and fungal menace that faces us. Okay, so what do we mean by a yeast cell? This is important. A yeast cell is a single cell with a single nucleus until cell division, and it grows by an asymmetric form of cell division called budding. Okay? And each of these cells now give rise to more cells, and each of these cells give rise to more cells until you have either an infection or, if you're growing it on agar, a colony, just like you get in bacteria. And because of the surface components of yeast cell walls, they absorb water and are nice, shiny, and pasty, just like bacterial colonies. And some yeast even have capsules that can produce a mucoid-like colony. Okay, when we talk about molds, okay, we are talking about a filamentous thing called a hyphae, or a hypha, singular, that grows, okay, and it often branches. So hyphae are very frequently branched. And the hyphae produce spores. Okay, these spores can be asexual, and they represent the way of sort of a modal stage of fungi, in the sense that they can grow over enormous areas. So here we took a single spore, planted it in the center, and let it grow for, in this case, probably about a month. And you see that it covered the entire plate, which is 20 centimeters. So this is 20 centimeters of movement. But it's been documented on an island off of Michigan that a single fungal mycelia grew in the ground over a course of 7 miles. So it's basically unlimited growth and unlimited movement. Considering the other thing about this fungal mycelia and the presence of these spores is that the spores can be blown in the wind, and being blown in the wind is another form of motility because, like pollen and other things that are blown in the wind, this thing moves them from one place to another where they can find other places to grow. Now, as you know, fungi are nature's recyclers. They are what causes, if you're walking in the forest, this is taking a lovely walk in the forest as opposed to one where you're avoiding Lyme disease, but taking a nice hike in the woods, and you see those charming, beautiful fungi growing, what they're doing is they're rotting that wood and recycling it back into the environment. They have lots of enzymes that produce, take polymers, organic polymers, and turn them into monomers, which is what they'll eat. And I'll tell you more about that later. Okay, now fungi, first and foremost, unlike most of the pathogens you've heard about so far this year, fungi are eukaryotes. That means they have a nucleus. Here's that nucleus, and the nucleus has linear chromosomes. They also have mitochondria, vacuoles, vesicles, a cytoskeleton, actin, myosin cytoskeleton. Okay? They have linear chromosomes with centromeres and telomeres and a mitotic spindle. All of these can be targets. They have a cell cycle. Here's that mitotic spindle that has to be made in order for the cell to divide. Okay? They have multiple RNA polymerases, just like we do. They have ribosomes that have bigger subunits than bacterial ribosomes that more resemble ours. Okay? Accordingly, they're more like us and they're harder to kill than bacteria. Okay? Antibiotics usually don't work, or the antibacterial antibiotics don't work. Don't forget that fungi make a lot of the antibiotics that we know about. Penicillin is a fungus. Okay? In addition, they do have a couple of differences that we can exploit with respect to chemotherapy. One is that they have a plant-like rigid cell wall comprised of a glucan and a mannan. I'll show you that in a minute. In addition, their cell membranes are slightly different from ours in that they have a different steroid content, with the different sterile content. So here's that cell wall, and that cell wall has 2 layers, an inner glucose polymer known as a glucan, okay, and that coming off of that glucan is a mannose polymer, shown here, otherwise known as a mannan. So glucan inner coat, mannan outer coat, and you'll see that this is an important chemotherapeutic target. Okay? The other important chemotherapeutic target is the fact that whereas we have cholesterol in our membranes, these guys have a slightly different sterol in their membranes. And don't forget, the bacteria don't have sterols in them at all, except for, good, remember, now I feel good, you remember that, mycoplasma. But these guys essentially make their own sterols. They have a whole sterol biosynthetic pathway. And instead of cholesterol, they make ergosterol, which, as you can see, has a major organic difference in the structure here in the form of this non-saturated bond. Okay? But that's very important with respect to chemotherapy, as you'll learn. Okay, now, the molds produce a mycelium. So this is what we call a mycelium. And that mycelium, as I mentioned before, is made up of hyphae, these tubular structures, as well as spores. And spores come in different flavors, as we'll see. Okay? And sometimes spores will be called conidia. Okay? And fungal classification is largely based on hyphal and asexual spore morphology. In most cases, what you're looking at in a infected tissue is you just see hyphae, okay? Or you'll see some other form of either hyphae. You might see yeast. Okay? In most cases, you won't see the whole fungal life cycle because that is not the right conditions for the whole fungal life cycle. But in general, what you'll just see are the hyphae. Okay? But in order to classify it, you very often have to grow it and see what kind of spores it make. And that leads us to a little bit of fungal morphology. We describe some fungi as being septate, which means that these sections of hyphae have little walls in between them. Okay? And then other fungi are what we call non-septate, which of course don't have those walls. Okay? And in these chambers here, or in this long tube, in all cases, with respect to hyphae, we're dealing with the syncytium, meaning multiple nuclei, whereas a yeast cell only has a single nucleus until cell division. Okay? So here's that syncytium, and it's got multiple nuclei. And yeast cells, on the other hand, have only single nuclei. Now, sometimes the yeast cells grow in chains, in which case only the terminal cell is the one that's dividing. And when that happens, we call that, instead of a hyphae, we call that a pseudohyphae. So when yeast cells grow within chains, they produce pseudohyphae or pseudomycelium. Okay? And again, each one of these yeast cells growing in a pseudomycelium has a single nucleus, except this guy on the end, which is dividing to produce two elongated yeast cells. So again, hyphae, pseudohyphae. Syncytium, single nucleus. Okay, mycelium, pseudomycelium. Okay, now spore morphology. Here is the language and the lesson. We're going to have a little bit in Greek. Okay, hyphal spores have different names that we have to know about when we're looking and describing them. So, for example, we'll hear about certain skin fungus having olorospores when they're grown usually in agar. These are what we call macroconidia. Okay, many times we call all of these kinds of spores I'm going to tell you about conidia. Okay, it's a general term for asexual spores, but specifically these are called macroconidia, and they're called olorospores because to the person who described them, they look like the aleuron on a wheat plant. So these are called olorospores. We'll see those when we talk about skin fungi. Arthrospores, arthros of course means joint, and these look like jointed structures. And arthrospores are spores that develop inside the hyphal wall, inside a hyphae, and they give that jointed appearance. Okay. Conidia, some, for example, aspergillus and penicillin, we call these little asexual spores conidia, and it comes after the Greek conus, which means dust. And if you've ever seen an aspergillus, it produces all this dust or a puffball, and each one of those little dust particles is a little spore. You can see them here. Okay, chlamydospore means thick-walled, thick coat, if I'm not mistaken, and these are round, thick-walled spores, and we'll see those when we look at Candida. We'll see those again when we look at histoplasmosis. Here is a chlamydospore, a thick-walled, big round spore. Finally, when a hyphae produces a yeast cell off of it, okay, when a hyphae produces a yeast cell, we call that a blastospore. Okay, and hyphae grow at a tip, shown here, or produce spores at their tip. You can see all of these things occur at a tip in most cases. Okay, we'll also hear, when we talk about coccidioidomycosis, about a structure called the spherule. And of course, we'll also be talking about yeast cells, which look a lot like the thing that we make bread and beer with. But of course, when we talk about them, we're going to talk about some dangerous yeast that are capable of causing serious disease. Okay, now we get to talk about sex. This is our sex ed portion. And sex in fungi, fungi have sex, okay? If we don't know about the sex of fungi, it's just because we don't know enough about the sex of that organism. And In most cases, in all cases, they turn out to, just like us, have two sexes. These can be called A or alpha, plus or minus, male or female. Doesn't really matter, and you don't have to worry about them. But the sexual stage of all fungi is how we classify them in the biota with respect to if they produce spores in an ascus, they're called an ascospore. If they produce spores that's on a base, they're called the basidiomycetes. So this is called an ascomycetes. This would be called a basidiomycetes. If they produce these big egg-like spores, they're called zygomycotic infections. And finally, if we don't know about them, they're called deuteromycetes. Okay, so the sexual cycle is basically this. For virtually all of them, we start off with a pink and a blue guy. You can assign whatever sex you want to either one, but they're generally growing and producing hyphae or yeast cells, depending on what kind of fungus it is. And essentially, if they grow, if two cells grow near each other, they're each emitting pheromones, okay? And opposite sexes secrete pheromones that induce a sexual cycle. And when they secrete those pheromones, what they're saying is, you're my species, but not my sex, so let's get together. And I write this down, you make my RAS protein go crazy, because essentially what it is it's a RAS protein-dependent protein exchange mechanism that stimulates a kinase and a kinase. Nothing you really need to know about, but a lot of the things that you learn about tumors and tumor genesis came from our understanding of sex and fungi, where we were able to characterize these kinase cascades that won Paul Nurse and Lee Hartwell and Tim Hunt the Nobel Prize. So it's just an interesting bit of factlet. And when that happens, when these pheromones stimulate the cells, they stop growing and they grow a new kind of cell that is that grows in the direction of the other fungus. The cells fuse and you form a zygote. Okay, now that zygote can sometimes grow and grow vegetatively, but ultimately that diploid form undergoes meiosis. So we have a classical meiosis with, if you remember meiosis, you get meiosis 1 followed by meiosis 2, produce 4 cells, and 2 of them are the blue sex and 2 of them are the pink sex. And when these guys germinate, we recapitulate the process again to produce more cells. Very simple sexual cycle, not unlike ours, except probably more haploid growth than we do, since our haploid cells in half of us have no growth phase. Okay, so here are some examples of the different fungal classifications. And this is something you just need to know because they'll refer to certain fungi as an ascomycetes, okay, which means that they produce spores in a sac. It's all you need to know. Here's a list of some of them. Okay, other fungi and mushrooms in particular are known as basidiomycetes. And here's a couple of them that we're going to mention today, though we're not going to talk about porcini, though we can think about having a wonderful pasta con funghi, which is one of the most delicious Italian things with porcini mushrooms. How many of you have had porcini mushrooms? Good. Best thing in the world. Almost as good as truffles, not quite. Don't forget, truffles are a mushroom. So that's why, another reason I love fungi. Okay, zygomycetes. Okay, and here's a picture of some mushrooms. Zygomycetes are these non-septate. These are all the non-septate fungi. They have a hyphae that is in fact very large in diameter. It's about 5 times wider than the hyphae on Basidio and Ascomycetes in most cases. And as I say, they produce a distinct zygote, like this big thick-walled zygote when there's mating. Okay, and we'll hear about a dangerous zygomycetes called mucor later on in the second hour. Finally, there are deuteromycetes, which are also called fungi imperfecti. And again, here's a case where we just haven't discovered the sexual cycle. Probably it exists. Sometimes it's imperfect because it may have mutated away, so we have to find that sexual cycle in a relative like we had to do with Candida. Candida albicans is an interesting fungus. We know it once had sex, but it mutated some of those genes away. So at least in our pathogenic species, we don't think they have sex anymore. They just grow via cell division. Okay, now, as I mentioned before, fungi are nature's recyclers. They degrade and live on dead organic matter, but as a result, some of them can actually still grow on living matter if there's no immune system trying to get rid of them. Okay. Now, many can actually live if you feed them a little simple diet of glucose, ammonia, sulfate, phosphate, and trace elements. Okay. And that's because what they're doing here is they're taking those polymers, secreting degradative enzymes, and turning them into monomers, ammonia, sulfate, phosphate, etc., which they then take up and recycle and use to make their own body. Okay. Now, in the lab, what we do is we routinely grow fungi on Sabouraud's agar. This is simply almost a cannibalistic like agar. It's got a protein hydrolysate known as peptone, which is essentially just a organic protein mix that comes from the slaughterhouse that you treat with peptic enzymes, with stomach enzymes, and has lots of almost everything in it, glucose and agar. So Sabouraud's agar is an essential thing you need to know about. That's our reference agar for classifying fungi. Okay, now when fungi grow, as I mentioned, they're eukaryotes. Okay, so they have all the things that we find in our own cells, like microtubules. They have chromosomes and nucleolus, nuclear pores, a actin cytoskeleton that they use for transporting things. And as most eukaryotics do, when they have to secrete something, they produce messenger RNAs that bind onto ribosomes that then secrete the proteins that need to be secreted directly in the lumen of the endoplasmic reticulum, like we have here. These ER then go fuse with the Golgi, and we have vesicle sorting, so you can see all these different kinds of vesicles coming out of the Golgi. And at this point, they're transported on this railroad system of most probably actin, but possibly in some fungi, some microtubules as well, but certainly in actin, to the cell surface. And what's secreted in most cases are two types. One is cell wall components and cell wall synthesizing enzymes that produce that bilayered glucan and mannan cell wall. So for example, all the mannan synthesis is done outside the cell, okay, after the secretion of the glucan cell wall. Okay? And more importantly, we also have vesicles that secrete proteases, nucleases, lipase, carbohydrolases, a nice mixture that if we took that on any living tissue, it would be just take whatever living tissue that is and turn it into a hydrolysate of gunk and gooey stuff and small molecules that if I were a fungus, I would then take those small molecules back up and resynthesize them into all of these things. So look especially at proteases. These can be invasive when it comes to pathogenesis, okay, as with the help of lipases certainly, and carbohydrolases. Now, an important aspect of fungal growth is the fact that fungi have different phases depending on the environment. And we speak of this for certain fungi as having what's called classical temperature dimorphism, in which case, if it is growing in the body at 37 degrees on a rich medium like blood agar without any immune cells to try and gobble them up and get rid of them, okay, 37 degrees, they'll grow as a yeast cell and produce, if I were to culture these yeast cells on blood agar, a typical pasty yeast-like colony. In contrast, if I were to chill these cells down and or put them on a less rich media than blood, like Sabouraud's agar, so either or, 25 degrees or putting them on a lower level of nutrients, like Sabouraud's agar, instead of growing as a yeast cell where they're happy as clams, just growing in place, they say, I got to get out of here. I'm going to grow and extend over a long distance and go search for a better environment, in which case they send out these tubes. And I told you these tubes can grow to unlimited lengths. And furthermore, they also produce these spores that are air stable and can be blown in the wind to even other environments where they can also plant themselves and try and grow. And if they land back in 25 degrees or Sabouraud's agar or not such great things, they'll produce a mold. But if they land in a 37-degree area, they're going to germinate and just grow as a yeast cell because they have a very rich environment to grow into. Now, this is called classical temperature dimorphism. We'll hear about some other kinds of dimorphism, and we'll also hear that sometimes the spores that are produced are of multiple types, okay, in the same fungus. Okay, so when we are diagnosing fungal infections, we still mostly do it via observing the fungus in infective tissue. This can be done in the case of skin, if we're looking at skin fungi, by treating the skin with sodium or potassium, usually potassium hydroxide, 10% for about 10 or 15 minutes. And what that does is KOH, virtually all the polymers that we make are soluble in 10% KOH, but fungal cell walls are not soluble in it. As a result, all the animal material, like the skin and the keratin, dissolves and we can't really see it. And you can see the leftover, I mean the fungi are dead in 10% KOH, but you can see the remnants of that cell wall, which makes it easy to see in the microscope. And usually this is observed at 10x, but you can even see it at, you can see it in most cases on 10, certainly 40x in a skin infection. Sometimes you make it a little bit easier in the sense that there's a stain called calcifluor white that binds to chitin in some fungi, not all, but many fungi have chitin in their cell wall. And if they have chitin in their cell wall and you add calcifluor white, this fluorescent dye will bind to the chitin and make the fungus fluoresce in a fluorescent microscope. When we're looking at cerebrospinal fluid for Cryptococcus in most cases, or histoplasmosis, we'll essentially spin down the cerebrospinal fluid so we can concentrate it and look at the precipitate and add a little bit of India ink and look under the microscope. And if we see yeast cells with a halo due to a encapsulated a capsule, we'll say it's Cryptococcus. If there's no halo in the yeast cell, we'll say it might be histoplasmosis. So India ink preparations are very important. And again, the halo indicates Cryptococcus, and no halo would probably be histoplasma. We can also use methanamine silver stain, which stains some proteins that are on the outer cell wall of these fungi, and they stain black. And you'll see that. Finally, gram stain, PAS stains. There are a number of different stains that Dr. Klein's going to tell you in addition to these. But make sure you know about 10% KOH and India ink with respect to fungal skin infections and CSF, cerebrospinal fluid examinations. Another thing about fungi is some fungi will actually fluoresce ultraviolet light. So if you take a long wavelength ultraviolet light, which is known as a Woods lamp, named not after Ed Wood, the famous film director, but after Dr. Woods, who found this out, you can actually observe some hair infections simply by shining a long wavelength UV light. Maybe if you ever get a job in a spook house where they have lots of ultraviolet light, you can count the number of kids with ringworm of the scalp, but I've never tried it myself. Okay. Now, I already mentioned this as a chemotherapeutic target, and I just want to mention it one more time: cholesterol in us, ergosterol in others. And this is a bunch of the antifungals that you'll hear about in your lecture on antifungal and antiprotozoal lectures. And I'm just going to skip through these. You can look at them for reviewing what Dr. Howland's lecture is about, but I'm not going to do it. But I do want to point out that this is a summary of the drug interactions and how they work for when you cover that in your next lecture. Okay, so now we're going to talk about specific disease-emitted fungal infections and taking into account what we learned already. And we're going to talk about tinnias or ringworms, Favis, a little bit about black palms, and pedro. Okay, now tinea, as you know, as we heard before, it means worm. Okay, it means thin filament, basically, in Greek. And initially, fungal infections known as ringworms were called tinnias because when they looked at them in the microscope, first of all, they were round on the surface, and in them there were these little filaments, so they got known as ringworms. So if you look at this fungal infection, you see these rings, and if you look in the microscope at the skin, you'll see hyphae. So they mistakenly called them tinnias, which when you hear your parasitology lecture, you'll also hear about tinnias, which are also worms. Okay, so these had thin filaments, okay, that are tinnias as opposed to tenias, and you'll get that when you learn about parasites. But ringworms are wherever you see these inflammation. If I were to take a skin sample from here, I would find little hyphae. I wouldn't find spores in almost all cases, but I would find hyphae if I were to do a 10% KOH. Now, tinnias or ringworms are named by where in the body they infect. So tinea capitis is ringworm of the scalp, tinea corporis, ringworm of the body, tinea pedis or athlete's foot is ringworm of the foot, Barbie, beard, tinea unguium of the nails. This is also called onychomycosis. Okay, and this is called jock itch. Okay, so as I said, if you were to do skin samples of the inflamed area here, or here, or here, and take a KOH, you would see hyphae. Okay, all of these tinnias that I showed you here can be caused by a fungus that belongs to one of these three genera: Trichophyton, Epidermophyton, and Microsporum. And you should memorize these three genera: Trichophyton, Epidermophyton, and Microsporum. Okay. As far as the species names, it's not very important. You may hear Trichophyton mentogrophites, flocosum, or odinii, or canis. You may hear them. The important thing is know that these are all fungal names, and if you have a tinea, you have a fungal infection, and it's a eukaryote. Now, by the way, this is important. In your syllabus, in your lecture note, is a table that I've assembled that have what I view as the essential fungi for you to at least memorize the names of, and again, more important, the genus rather than the species. So look at those tables if you need a review to make sure that you know enough for your exams or your boards. Okay, so that should be at the end of lecture 1 in your syllabus. Okay, so all of these genera are likely to cause any of these, but in most cases of athlete's foot are caused by Trichophyton mentogrophites. Most cases of tinea capitis are caused by microsporum, for example. Not entirely all of them, though. Okay, this is not what they look like, but they do look, when they're growing on agar like this. Here are two species of trichophyta. Again, you see this mold. And the important thing I want to show you about this slide is that these genera, all the Trichophyta and Epidermophyton and Microsporum, all are very slow growing. It takes about a month for them to grow out on a petri dish like this. And we make them grow a little bit faster in about a month by adding a little bit of olive oil to our media. Now, if you saw 60 Minutes last night, I don't know if it's extra virgin olive oil that you have to add. It's just lipid. Okay, so and I don't know whether the food mafia is involved, but you have to add lipid to this. And most importantly, none of these genera grow well, or really grow at all, above 34 degrees, which is good because why? Because they can't grow within us, but they can grow on our skin, which is which is a little bit colder due to ambient temperatures. Very important. Okay, so here is why they all look alike. They all produce macroconidia that are classified as allospores when they're grown on sabrodes. In most cases, growing within us, you'll just see fungal hyphal tubes without seeing any spores. Okay, so how do you get athlete's foot? We all have been given the lecture about wearing shoes in the locker room. And it's actually true, because what happens when you get athlete's foot, in most cases, is that a piece of skin is being degraded by that fungus. So here, the fungus is growing on the foot, and as you can see, it's causing the skin to flake off. And those little skin flakes from somebody who had athlete's foot might be all over a locker room floor, and is likely to get lodged in between your feet or on your nail bed over here. And when that happens, you can get, if it's allowed to stay there, that fungus from that little bit of skin can grow into your skin and cause that infection. Okay, now, if it gets in the nail bed, that nail is infected, and you'll see that that this fungus would actually grow in all directions and ultimately infect the nail bed. And if what happens here in a fungal infection is that the fungus is secreting proteins. Those proteins cause an allergic type 2 hypersensitive reaction, which of course causes you to itch. Okay, what happens when you itch? You scratch, exactly. When you itch, you scratch, and if you scratch, you help scratch off what? Skin, which essentially is part of the ecology of these fungal infections that helps them spread themselves around the biota. Okay, now if you scratch so much, you can do tissue damage and get secondary infections and horrible inflammatory responses that are due both to the fungus and other bacteria. So these are usually super infections of just more than the fungus. Okay, another thing that happens, here's another case of a little boy with tinea capitis, and he's scratching because it itches, and he's a little kid, and so he scratches and he produces this open wound which has this oozing like lymph fluid that comes out of it that's, in the case of a fungal infection, referred to as carrion. Okay, if you get an infection of your nails, as I mentioned, it might start over here and then ultimately grow back to the nail bed. Here's an interesting case in the sense that the nail is being degraded as it's growing. The nail's being degraded as it's growing, and you can see the nail degrades, and he can't grow long, beautiful nails, so he can go and get himself a manicure because it's rotting beforehand. Another thing that happens very often in what I call, refer to as old geezer nails, is that, and you've seen them, I can show you mine, but you don't want to see them. What happens is that it causes a hyperkeratosis. It actually stimulates and makes for a very thick nail that grows out of it. And that nail is almost porous by the time it reaches the base of the front of the nail. So you have this growth of the nail and the growth of the fungus as it grows out. You see that the nail is a little healthier just because the amount of fungus is less than it is here. The fungus is growing. This is an important aspect of the treatment, and why nail fungus is so hard to deal with is that until recently there were no drugs that perfused into the nail bed. Now, if you watch television late at night, you'll see a prescription antifungal imidazole that actually is able to penetrate into the nail bed. But in either case, so in the old days, you used to take systemic antifungals for about 6 months so that you do a whole cycle of fungal growth to make sure that all the nail has grown out without any fungus. Now you still have to do a whole cycle of treatment with that imidazole that penetrates here. So that becomes more difficult. And since these things are just simply ugly and really are painless and have no other symptoms, in most cases, old people just ignore them, just have ugly nails that show up at the beach and at resorts, and you look at them and go, God, what a disgusting bunch of nails. I hope I never get them. But in most cases, as you age, it becomes a fairly common problem. Okay, so that covers those 3 genera, but there are a couple of other species of fungi that cause skin infections that we should cover. One is Malassezia globosa and Malassezia furfur. This causes what we call tinea versicolor, ringworm that changes color. And it changes color because when you have an infection of this and you tan, the contrast increases. The other thing is that the fungus kind of inhibits melanin production, so you end up with these light spots where you are infected and your skin's a little bit darker. And if you tan, it becomes even more contrasting. Okay, another fungus that I'm not even going to mention the name of produces what's called black palms. This is only found in tropical regions, and this is because virtually all of these organisms are found in the environment, growing in the soil, rotting organic matter, and this one is found in the tropics that just produces an asymptomatic darkening of the skin known as black palms. It can virtually infect anywhere. Another species of trichophyton called Trichophyton schoenleinii, instead of producing flakiness, actually produces a similar hyperkeratosis like we observed in the nails shown here. And so you get these scales, these favic scales. And that disease is called favus. Again, it's largely, other than cosmetically, it's painless. It doesn't really hurt, but it is somewhat disfiguring. It can cause hair loss growing on the scalp and these hypertrophic scales. Okay. Finally, you can get fungi that just infect the hair shaft. One of these is black pedra, which grows up as a black mold, and another one is white pedra, which grows up as a white mold. And here you can see arthrospores, okay, and that grows on the hair shaft. These can be dealt with simply by cutting the hair, and that rids us of the infection because they are exclusively growing on hair. Okay. Very quickly, all of these skin infections are again diagnosed by treating whatever you think is infected based on that redness with KOH and looking in the microscope and finding hyphal tubes that can, of course, have different morphologies depending on the particular fungus that's infected, not this guy. And again, that Woods lamp. I'm just not going to show you that. Finally, we'll just conclude this part of the lecture. Some people are highly allergic to fungi, so if they get a skin infection that produces antigen that can leach into the body, it can produce a systemic allergic response known as the dermatophytid response, or simply id response, that can produce lesions, basically hives, urticaria type lesions all over the body. And again, if you were to look at these lesions, there'd be no fungus there. But if somebody showed up with this, you might want to look all over their body for a fungus infection of the skin that produces this dermatophytid response. Okay, and this is just, again, the antifungals, a list of the common antifungals, which we're not going to talk about. And then in the second hour, we'll go a little bit, so we still have 5 minutes, so let me just start and talk at least about one subcutaneous mycotic infection. And that being, so these infections, you need to be stabbed by the fungus in the dermis, okay? Below the epidermis, these can in some cases go systemic. They can certainly grow in, but they usually grow initially, and in most cases, at the site of the lesion where they've been stabbed in. So let's talk about sporotrichosis first. This is the first classically dimorphic fungus that we're talking about. Okay, Sporotrichum or Sporothrix schenkii. It grows on Sabouraud's agar, shown here, as a mold. And in the microscope, if you were to look at this, you'd see hyphae with blastospores growing off of these hyphae. Okay, but if you put it at 37 degrees on blood agar, it grows as an elongated yeast cell, shown here. Okay, so blood at 37. Okay, which means that in our bodies, if we were to find an infected lesion with this fungus due to stabbing, okay, we would find the yeast phase, shown here. Okay, so this is what sporotrichosis looks like. Okay, and here is a result of probably 2 or 3 stab wounds. It's also called rose thorn disease. It's a ubiquitous fungus that's found on lots of plants and in the soil. And if you get stabbed by a thorn, you might get this very painful lesion. Okay, now sometimes the fungus can actually invade the lymphatics and cause eruptions up and down the lymphatic systems. So this is referred to as a lymphocutaneous infection. Okay, and we could probably find hyphae growing all through the lymphatic and causing these eruptions here. Or it can produce what we refer to as a fixed cutaneous lesion, sporotrichosis. Very painful, a lot of tissue damage that causes great pain. If we were to look at the pus from one of these lesions, we would see these elongated yeast cells. If we stain with silver, they would light up very easily. And you see just yeast cells. They don't grow that fast, so we don't see many buds, but we can see some cells with buds if we look hard enough. This is, again, Sporotrichum schenkii or Sporothrix. Okay, this is a famous New Jersey case a couple of years ago. A 65-year-old New Jersey man developed a large sore in his genital area that refused to heal despite dozens of tests, 7 medications. Turned out he had sporotrichosis, a fungal infection from plants. He probably picked it up when he urinated against a bush at his favorite golf course. Okay? And again, sporotrichosis is sometimes called rose thorn disease for obvious reasons. Okay, let me just finish with the 2 most disgusting slides. Another subcutaneous infection can be caused by many different kinds of fungi, among them Pseudo-Alicaria, Magirella, Exophiala, and common Aspergillus. If it gets poked down deep and you have a slightly immunocompromised state, you can develop these horrible edema-like lesions. You'll hear more about edema coming from fungus from Dr. Klein's pathology lecture next week or later this week. And what happens in this case is the fungus basically grows from the skin down and occludes the lymphatic, causing this edema and causing this horrible like lesions. Obviously, this is a case of incredible neglect. In most cases, you might see one lesion or two lesions. This is somebody who clearly didn't go to the doctor. It comes from, the name Madura foot comes from the fact that it was initially characterized in Madras, India, hence the name Madura foot. Okay? It's more commonly referred to as mycetoma, due to these big growths that occur in the region that is infected. Okay? And you might want to also remember pseudoallocharia as a name and exophiala as a cause of these, the more common causes of these lesions. Okay? Now, very often these lesions are superinfected with actinomycetes, and it's important to characterize the organism that's causing the lesion, because if a mycetoma is caused by an actinomycetes, it's a bacteria, and it needs to be treated not as a fungus but as a bacteria with conventional antibiotics. Okay, finally, chromomycosis, as the name implies, chromo or dark colored, produces this dark pigmented yeast. These species, these genera, Fonseca, Cladosporium, and Phylaphora, all are capable of causing these cauliflower-like lesions that are due to stabbing and then growing within and on the surface of the foot. Now, in general, a case of chromomycosis is going to look like this, not like this. This is another case of incredible neglect. In all cases, these need, because you can't get antifungals into a lesion like this in order to cure it, they have to be treated via surgery, surgical removal. In this case, a lesion like this isn't a big deal. To remove a lesion like this might cause the loss of this guy's foot. Okay, now these fungi all produce these spores that can usually be observed in these fungal infections. They're called sclerotic bodies, but they are round spores, possibly sexual spores. This almost looks like an ascus. And if you were to grow this and look in the microscope, you would see hyphae with spores. Okay, so that concludes superficial and subcutaneous mycotic infections. We'll begin the second lecture talking about systemic infections in about 7 minutes. So we'll begin at 2 minutes after. Thanks. Okay.